Good morning, good afternoon, or good evening. This is Lisa Whitehead, and we are studying the brain and cranial nerves, which is in the course of Biology 205, Anatomy and Physiology 1 at Illinois Central College. Today's lecture includes the following learning outcomes: to identify the major regions of the brain and describe their basic functions. This includes the cerebrum, cerebellum, diencephalon, mesencephalon, or midbrain, the pons, and the medulla. Also, to understand the embryological origin of the adult brain structures as shown in Table 14.1. Also, on a diagram or on a sheep brain as per lab, to identify the brain structures listed in the Lab 11 objectives. To name the ventricles of the brain and the connecting pathway. This includes the interventricular foramina, the cerebral aqueduct, the median and lateral apertures, and then understand their functions. To explain the function of the meninges of the brain and to locate and explain the function of the dural folds. Describe the processes involved in the formation and function of the cerebrospinal fluid, or CSF. List the pathway of CSF, starting with production and then going into entering the venous system. And to label the blood vessels going to and away from the brain. This includes the carotid and vertebral arteries and also the jugular veins. Also, you'll describe the blood-brain barrier, abbreviated BBB, that astrocytes form it, along with tight junctions in the capillaries, and then what crosses the blood-brain barrier. To explain why there are breaks in the blood-brain barrier at the hypothalamus, at the posterior pituitary, pineal gland, and also the choroid plexus. To describe the anatomical differences between the medulla oblongata and the spinal cord. To list the main structures and functions of the medulla oblongata, the reticular formation, the 5 cranial nerves attaching here, and relay stations, and the relay stations specifically will be covered in this lecture. And then to list the main structures and functions of the pons, the 4 cranial nerves, the respiratory rhythmicity centers, nuclei, and tracts. And to list the main structures and functions of the cerebellum. This includes the folia, the vermis, the cerebellar hemispheres, arbor vitae, Purkinje cells, and peduncles. Also, you'll list the main structures and functions of the midbrain, or mesencephalon, and this includes the corpora quadrigemina, comprised of the superior and the inferior colliculi, the red nucleus, substantia nigra, peduncles, and reticular formation. You'll know to list the main structures and functions of the diencephalon. This includes knowing the pineal gland of the epithalamus and the function of melatonin. To define the role of the thalamus, and this excludes the 5 specific groups. You'll explain the 8 functions of the hypothalamus, explain the function of the pituitary gland and know what the infundibulum is and where it is, define the role of the limbic system, and to explain the 3 types of axons that are present in the cerebrum and what their functions are. So this will include association fibers, the corpus callosum and the anterior commissure, and projection fibers. You'll define the function of the basal nuclei, describe the anatomical location and function of the 4 lobes of the cerebrum, to label a diagram of the motor and sensory areas of the cerebrum, including all 4 lobes being the frontal, parietal, temporal, and occipital lobes. The motor areas, including the prefrontal area, the premotor area, the primary motor cortex, Broca's area, and the central sulcus. Also the sensory areas, including the primary somatic sensory cortex, the somatic sensory association area, Wernicke's area, visual association area, and the visual cortex. And then for the special senses, the visual cortex, the visual association cortex, the auditory cortex, the auditory association cortex, the olfactory cortex, and the gustatory cortex. You'll also describe the functions of the motor and sensory areas of the cerebrum as mentioned above, and describe the processing pathways that would occur as signals are received, processed, and then sent out using both the motor and the sensory areas of the cerebrum. And so an example here that you'll be using will be the example of burning your hand on a stove. So you'll need to be able to understand and explain the pathways to 1, recognize that it is in fact a stove, to 2, realize that you're actually burning your hand, and then how to remove your hand away from it, pretending that your interneurons aren't involved here yet. And then 3, to know to say, ouch, to recognize the pain. Last, you'll be describing the significance of the volume of space taken by different body parts when looking at a homunculus of the primary somatic sensory cortex and the primary motor cortex. You'll explain what decussation is and where that occurs. You'll identify the main general functions of the hemisphere in the population majority. You list on a chart the details of the 12 cranial nerves. listing the proper name using the proper Roman numeral. You'll also define whether these nerves are sensory, motor, or both sensory and motor. You'll be able to recall their major functions, and we'll be using a number of mnemonics to learn this, which will be very helpful. And also, last, you'll be describing the cranial reflexes of corneal, auditory, and tympanic reflexes of the corpora quadrigemina, and also for the photopupillary reflex. In today's lecture, we're following our textbook again, so we're going to start off with section 14-1, which is brain development. We're going to proceed through the chapter looking at how the brain develops, then how it is protected. Then we'll discuss the brainstem, moving from the medulla oblongata to the pons and the midbrain. And then we'll move up to the cerebellum, the diencephalon, and then the limbic system, finally pulling it all together with our motor, sensory, and association areas of the cerebrum. Then we'll identify and discuss all of the cranial nerves, all 12 of them, and then finally wrap it up with a quick discussion about cranial reflexes. Today, we're going to be studying the brain and cranial nerves, and I think you're going to love this chapter. The adult human brain is fascinating. So the adult brain is going to have about 97% of our body's nervous tissue, which leaves the remaining 3% to be composed of our spinal cord, which is part of our central nervous system, and our remaining peripheral nervous system. The average weight of the brain is about 1.4 kilograms, which would equal about 3 pounds. And the volume is typically about 1,200 milliliters, and this will range in totally normal adults from 750 milliliters all the way up to 2,100 milliliters, which is almost 3 times as big. It's worth noting that the brains of males are actually about 10% larger than that of females, but there's absolutely no correlation whatsoever between brain size and intelligence. First, let's talk about the brain overall and what are the general structures we need to know about before we dive in a little deeper to look at the structures that are within the overall structures. So first, we have the cerebrum. That would be what we would normally call the brain. So the large component in the image that you see to the right, showing all of those convoluted surfaces, that is the cerebrum. And then the part underneath, that is that bluish shaded area that's more finely convoluted, that is our cerebellum, sometimes even known as our lizard brain. We're also going to discuss today the diencephalon, which is an important part of our brain that is basically up on the center inner aspect of our brain. It's deep to the cerebral cortex. And then further to that, as we go a little more inferior, we're going to look at the brainstem. And so this is where the brain structures will start to connect down to the spinal cord. The cerebrum is the largest part of our adult brain, and it again is what we think of when we imagine in our minds what the brain looks like. So the cerebrum is responsible for controlling all of our higher mental functions, and this includes things like our conscious thoughts, our problem solving, our intellect, our memory. All of these things are controlled by our cerebrum. So the first breakdown of our brain is dividing the cerebrum into left and right cerebral hemispheres. The outer surface of the brain is called the cerebral cortex, and the cerebral cortex is covered by a surface layer of gray matter. And as you'll probably recall from our previous chapters, gray matter contains neuron cell bodies, whereas white matter contains neuron axons. So when you look at that outer surface of the brain, you're going to notice all of those convolutions. So we're going to describe them in one of 3 ways. They're either a shallow groove, a deep groove, or a rounded eminence sticking up. So any of the rounded spots that stick up are called gyri, or singular gyrus. And if it's just a shallow demarcation, then we'll call that a sulci or sulcus singular. And if it's a deeper one, we'll call it a fissure. Think of like a tear, like a fissure. So fissures are used oftentimes to delineate one region of the brain to the other. The cerebellum is the second largest part of the brain, and it's that bluish area that you see in the image to the right. So it is basically called our reptilian brain because it's responsible for some very primitive responsibilities, like repetitive body movements, for example. There are 2 hemispheres in the cerebellum, just like there are in the cerebrum, and it's also similarly covered with gray matter. In this case, we call it the cerebellar cortex as opposed to the cerebral cortex. Inferior to the cerebrum and anterior to the cerebellum is the diencephalon. So the diencephalon is a region of the brain that contains both the thalamus and the hypothalamus. The thalamus is important for relaying and processing sensory information, and the hypothalamus has responsibilities for emotion, autonomic function, and hormone production. Within the diencephalon is another structure called the pituitary gland, and this is our major endocrine gland. In fact, it's often known as our master gland because it's responsible for integrating our nervous and endocrine systems, so it really has a lot of control in our body. So as you can see, it's kind of dangling off the front, but it's connected up to the brain via the infundibulum, which is just another name for the stalk. Proceeding inferiorly, we have the brainstem. And the brainstem includes the midbrain, the pons, and the medulla oblongata, which if you ever watched Billy Madison, I'm sure everybody is saying it in a very specific voice in their head right now. But the brainstem is really important for relaying information between the spinal cord and up to the cerebrum or the cerebellum for processing. The midbrain is highlighted in the image on the right in green, and it's labeled as letter D, and you can see it just kind of tucked in between the diencephalon and then the structures below. So the midbrain is responsible for processing sight, sound, and our associated reflexes to sight and sound. And it's also responsible for maintaining consciousness. The pons is just inferior to the midbrain, and it's kind of sticking out a little bit anteriorly. And the pons will connect the cerebellum to the brainstem, and it's going to contain tracts, which are just collections of central nervous system axons. It also contains relay centers for information, and nuclei for somatic and visceral motor control. And when we say nuclei in the context of the central nervous system, we're talking about places in which there are concentrations of cell bodies. And in these places, this is where a lot of processing occurs, and these are the control centers for a number of different functions. So we're going to discuss nuclei in a lot of different contexts coming up. But for now, just know that the pons includes nuclei for somatic and visceral motor control. The medulla oblongata is even further inferior to the pons, and it is the most inferior component of the midbrain. So it connects the brain to the spinal cord. The inferior portion of the medulla oblongata has a very narrow central canal, and that central canal is going to persist throughout the spinal cord as it proceeds down the vertebra. And that central canal, of course, has blood vessels in it that allow materials to diffuse out to supply the spinal cord with nutrients and oxygen and other gases, etc., etc. It's also responsible for relaying sensory information and regulating autonomic functions, which include things like our heart rate, our blood pressure, and also digestion. If the opportunity ever arises for you to study embryology, I highly recommend it. It's incredibly fascinating, and I think you would really enjoy it. And it all originates from the neural tube when we're discussing the brain and the cranial nerves. So the neural tube is the very most primitive part of brain development, and that tube is going to become everything that we have in our brain. And so the rostral portion of this is going to enlarge, and as it does so, it's going to form three main parts, which are called primary brain vesicles. There is the prosencephalon, the mesencephalon, or the rhombencephalon, and these are known as the forebrain, midbrain, or hindbrain, respectively. So you can see in table 14.1 down at the right that it shows you what a primary brain vesicle looks like at 3 weeks of development. Keep in mind this is 21 days, so it's very, very tiny and very primitive. And then as it expands and grows and twists, you're going to see some secondary brain vesicles develop by 6 weeks. And so the prosencephalon will develop into the telencephalon and also the diencephalon, whereas the mesencephalon, which is our midbrain, will form our midbrain of our head, and then further our cerebral aqueduct, which we'll discuss more when we get into the ventricular system later on. And then the rhombencephalon from the 3rd week will also become the medencephalon, which is the cerebellum and pons, and the myencephalon, which becomes the medulla oblongata. The ventricles of the brain contain cerebrospinal fluid, and they also produce and maintain that cerebrospinal fluid. So this occurs through the development of the embryonal neural tube, which will then enclose a neurocele, and the neurocele expands, and when it does that, it's going to form these sections called chambers, which we'll understand to be ventricles, and the ventricles are lined with ependymal cells, some of which are going to be specialized to be able to create and maintain CSF. So in each cerebral hemisphere, which is either the right or the left side, we're going to have one large lateral ventricle. So that means because we have 4 ventricles, the first 2, 1 and 2, are right and left. We don't number the right one as number 1 and the left one as number 2. We just call them the lateral ventricles, and those are the first 2 ventricles. So those first 2 ventricles are separated by a single wall in between them, which is called the septum pellucidum. And the septa, or septum, is a wall. And if you think of the word pellucidum, lucid just means clear, right? If somebody's thinking clearly, they're very lucid. And so pellucidum is a very thin, narrow wall that separates out the 2 ventricles. There's also the third ventricle that's in the diencephalon, and this one will communicate with both of the lateral ventricles through the interventricular foramen. And then we have the fourth ventricle even further inferior to that, and that's going to extend down into the medulla oblongata and communicate with the central canal of the spinal cord, thereby gaining access to the remaining spinal cord inferiorly. It also connects superiorly with the third ventricle through the narrow canal called the cerebral aqueduct. So if you look at the image on the right, which is figure 14.2 in your text, you'll see the ventricular system of the brain, and it's showing you the top 2 major large structures are the lateral ventricles, so that's 1 and 2. As you go inferior to that, CSF will come down through the interventricular foramen into the third ventricle, which is another collection site, and then it'll go down further into the cerebral aqueduct and drain down into the fourth ventricle and then down to the spinal cord. Figure 14.2 in your text shows you both a lateral view and an anterior view of the brain and how the ventricles are oriented. And from the lateral view, it looks pretty clear, but when you look at it from the anterior view, it almost looks like bull horns, because that's how curved those first and second lateral ventricles truly are. So just keep in mind that they are curved in that regard, because that can be misleading when you only look at a lateral view. So you're looking here at the superior lateral ventricles, and then draining down to the third ventricle through the interventricular foramen, so interventricular between the ventricles. And then to drain in between the third ventricle to the fourth ventricle, we have the cerebral aqueduct. And I think of an aqueduct as a large waterway, right? And so at this point, we have all of this fluid coming from superior down to the bottom, and it's going to go down to the spinal cord. So it's collecting down here in the cerebral aqueduct to expand out into the fourth ventricle before going down into the central canal in the spinal cord. Now let's discuss section 14-2, how the brain is protected. The brain requires a lot of protection because it's a very vulnerable organ. It's very soft and gelatinous, and you would think that it would be very firm, but in real life it's considerably softer than what you would imagine. And so we have physical protection to it, which includes the bones of the cranium, which you'll study in 206. There are cranial meninges, so those are connective tissue coverings that will hold the brain in place and anchor it. And then we also have cerebrospinal fluid, which basically acts like a nice cushion all the way around the brain. So the cerebrospinal fluid is more like an airbag, whereas the cranial meninges are more like a seatbelt, if you want to think of it in a car term. And then we also have some biochemical isolation that will protect the brain from everything else because it's so vulnerable. And this is the blood-brain barrier, which we'll discuss later on. But it's worth keeping in mind because as you study in future classes, you'll find, especially in pharmacology, this becomes very important because some drugs will cross the blood-brain barrier and others will not. So you'll notice that when you look at your pharmacology texts. Although we previously discussed the meninges, I'm just going to briefly review them here. So the cranial meninges are continuous with the spinal meninges. They're not significantly different. There are a couple adjustments which I'll discuss in a moment. But first we have the dura mater, remember the tough mother, and the dura mater has the inner fibrous layer, which is the meningeal cranial dura, and the outer layer, which is the periosteal cranial dura. And this would make sense because the outer layer is closest to the cranium, which is the skull, right? So periosteal, peri meaning around, and osteal meaning bone. So I think that's fair enough. And this is going to be fused to the periosteum, so it's not going to be easily separable. We also have arachnoid mater, which is a little bit closer to the brain, and then the pia mater, which is that very fine material right up upon the brain. So if you look at the superior part of figure 14.3, you're going to see a couple different things happening. At the very bottom of the image, you'll see the cerebral cortex. So that's the outer aspect of the brain, which is composed of gray matter. And then right upon the cerebral cortex, you're going to see that very fine layer of pia mater. And then there's that web-like area. Remember arachnophobia, fear of spiders? It's like a spider web network. So the subarachnoid space is right up next to the pia mater, and then next to that we have the dura mater, which is a bilayer material, right? We've got the meningeal cranial dura and the periosteal cranial dura, and in between those two we have the dural sinus, which we'll discuss next. The dural folds, which we just looked at in the previous slide, are those spaces in between the two layers of the dura, and they're extensions of the meningeal cranial dura into the cranial cavity. And they are there to stabilize and support the brain, but I think most importantly, you would imagine that they're there because they contain collecting veins, and these are called the dural venous sinuses. And so there are 3 dural folds where the dura is going to fold in, and this creates separations in the brain. Although the brain is all continuous, it does create just a little bit of extra support. There's the falx cerebri, the tentorium cerebelli, and the falx cerebelli. These 3 structures have a very important role in anchoring the brain and holding it in place. They also provide a lot of protection. So let's look at the image on the right first. So the broadest area that you see, the broadest dural fold, is the falx cerebri. And this is going to separate the right and left cerebral hemispheres. So in this, up at the very top, you're going to see the superior sagittal sinus. And then at the bottom of it, you're going to see the inferior sagittal sinus. Then you'll notice, if you move a little bit more inferiorly, right in between where the cerebrum and the cerebellum would be, there is a horizontal broad swath of material, and that is our tentorium cerebelli. And if you think of it, it's basically pitching a tent over the cerebellum. So what this is doing is separating the cerebrum from the cerebellum, and it's going to contain the transverse sinus, which runs transversely. We also have the falx cerebelli, which, just like the falx cerebri, which separates the right and left cerebral hemispheres, the falx cerebelli is going to separate the right and left cerebellar hemispheres, and it does so just below the tentorium cerebelli. So moving deeper to the dura is the arachnoid mater and the pia mater. So the arachnoid mater, which is that web-like material, it's going to cover the brain, and typically we're going to expect it to be attached totally to the dura mater so that you don't even really realize they're 2 separate layers. But it can be separated by a subdural space in the event of injury or illness, or, for example, a bleed. So that subarachnoid space is what it is that lies in between the arachnoid mater and the pia mater, which is 1 step further more deep. The pia mater is that very, very thin, fine layer that attaches to the brain surface, connected by astrocytes. Cerebrospinal fluid is going to surround all of our exposed surfaces of the central nervous system. And its function is there to just basically support the brain physically and to cushion our delicate structures against being smashed against things. So it acts like a big water balloon buffer around our brain within our skull. And it's also there to transport nutrients, chemical messengers, and also wastes. Cerebrospinal fluid is produced by the choroid plexus, which is including some specialized ependymal cells that are going to surround capillaries so that they have access to all sorts of different nutrients and fluids and gases. The choroid plexus is going to secrete about 500 milliliters or half a liter of cerebrospinal fluid into the ventricles every day, and it will also remove waste products from our cerebrospinal fluid and adjust the composition of it on an ongoing basis. So if you look at the image at the bottom, you're going to see one major projection down, which is the choroid plexus. And you can see that all of those cells, all of those specialized ependymal cells, are held together by tight junctions. And they're right up next to a capillary, so that way it has easy access to all sorts of fluids and materials that are coming through the capillary that they can either use to put into the CSF or use as an exit point to pull something out of the CSF and into the capillaries to be discarded as waste. We know that the choroid plexus creates cerebrospinal fluid. Now let's talk about how it circulates. If this is something that's confusing to you, take a moment to look it up online and find another schematic or a quick video or something to have this make sense to you. So the CSF starts at the choroid plexus where it's generated, and then it's going to go through the ventricles, and from there it goes to the central canal of the spinal cord, and then into the subarachnoid space via the two lateral apertures and one median aperture in the roof of the fourth ventricle. And in doing so, it's able to support the brain, the spinal cord, and that cauda equina. Remember, that's the horse's tail, the end of the spinal cord. So let's look at the image on the right. What we're looking at here is up at the very top, we have production of the CSF at the choroid plexus. So the CSF is going to go into the lateral ventricles, which is the first and second ventricles. They're paired, remember the right and the left. And then it's going to drain down from the top down through the interventricular foramina, which is foramina is a window, into the third ventricle. So once it's in the third ventricle, it's going to continue further south or further anteriorly through the cerebral water duct, or the major waterway, into the fourth ventricle. At this point now, it's going to go down through the lateral and the median apertures into the subarachnoid space, and then the arachnoid villi of the dural venous sinuses, and can filter out to the venous blood, so that way it can go down to the heart and lungs and get pumped back through our cardiovascular system into our arterial blood. So now we have fresh blood coming up back to the fourth ventricle's choroid plexus and the third ventricle's choroid plexus, and up to the first and second, or lateral ventricles, plural, choroid plexi or plexuses. Arachnoid villi are just extensions of our arachnoid membrane, but they're going to extend into the meningeal layer of the dura mater and further into the superior sagittal sinus. So these in adults can become arachnoid granulations, which you can see at the image on the right. So you can see it's this white, almost papillary, it's almost warty, right? It's varicoid. It's kind of a gross looking structure that you wouldn't imagine is normal if you were to first look at it. And so these are called arachnoid granulations, which are just large clusters of arachnoid villi in adults. They are totally normal, and they are there to absorb CSF into our venous circulation. So fun story for you. When I was studying anatomy at grad school, I remember looking at my cadaver, and I saw these arachnoid granulations before we had the lecture and before we read this content. And oh my God, I was convinced that this patient must have had some sort of metastatic transitional cell carcinoma from the bladder, because that was the type of tumor that it resembled to me right away. And I was very shocked by this whole thing. So then once I learned that they were arachnoid granulations and they were normal and they were on all of these different cadavers in the room, like all 90 of them pretty much, I realized, okay, yeah, that's truly a normal structure, even though it looks really alarming. A major function of the cranial meninges and the CSF is just to protect the brain, right? So it's going to hold the brain in position. Specifically, the dural folds really hold the brain in position and anchor it. And it protects it from damage that would happen if that brain were to slap up against the cranium on the inside. So we have CSF there, our cerebrospinal fluid, to cushion the brain against any sudden movements. Cranial trauma is what will arise anytime we have a head injury that results from impact with an object. So this could be classified as either a coup or contra-coup injury. Coup, spelled C-O-U-P, is when if you fall down and hit your head on your forehead and your brain slaps up on the inside of your brain against your forehead and you've got a bruise on the inside of your brain there, that would be a coup injury. But in really severe accidents, so let's say you're in a severe motor vehicle accident and you hit your head on the steering wheel and then your head slaps back in a whiplash sort of movement and you hit your head on the back seat. In that case, your brain is slapping around inside your brain, both forwards and then backwards also. So you have what is called then a coup and a contra-coup injury as well. Contra just meaning against or opposite. The brain is supplied by blood that comes up to it by the internal carotid arteries in our neck and vertebral arteries, which are more superior to that. And those two are going to deliver blood and nutrients and oxygen to our brain. Most of the blood that comes to our brain is going to be removed by the dural sinuses through the internal jugular veins coming back down inferiorly. Now, cerebrovascular diseases are any disorders that are going to be classified as those that interfere with the blood supply to our brain. So what you might have heard as a stroke is actually called a cerebrovascular accident. So that's abbreviated to the acronym CVA, which is how you'll chart it. And a cerebrovascular accident is when the blood flow stops arriving to a portion of the brain and then that portion of the brain dies. So the affected neurons are going to die within minutes of having inadequate blood supply. This can happen in one of two ways. You can have either a hemorrhagic stroke or hemorrhagic CVA or an ischemic CVA. So a hemorrhagic one is when you have a brain bleed. And so the bleed is just basically pouring blood into the area around the tissue, but not actually going into the place where it should be because of a rupture, usually typically an aneurysm. Now, an ischemic stroke is different because an ischemic stroke is what we're looking at in the image on the right, in which you see blood vessels that are occluded because we have either a blood clot or a piece of plaque that is ruptured and dislodged. And basically the same thing happens in the heart as is in the brain. With ischemia, we have a stoppage of blood flow in a blood vessel that is just proximal to that distal tissue, and the distal tissue will subsequently die because of a lack of oxygenation. So in the heart, we have a myocardial infarction if one of the coronary arteries in the heart has an obstruction, and if we have that same obstruction in the brain, then we have a cerebrovascular accident, specifically an ischemic cerebrovascular accident. The blood-brain barrier is an invaluable structure in that it's going to separate the central nervous system out from the general circulation and to keep it safe. And it is separated by a network of tight junctions between the capillary endothelial cells in the central nervous system. So typically what you would see is only lipid-soluble compounds like oxygen, carbon dioxide, steroids, prostaglandins, and even small alcohols are able to pass and diffuse into the interstitial fluid of the central nervous system. But astrocytes are there to regulate the blood-brain barrier to allow some things to pass and others not to. So ultimately what astrocytes do is they release various chemicals that will control the permeability of the endothelium. The blood CSF barrier, which is different than the blood-brain barrier, is formed by specialized ependymal cells. They're connected by tight junctions. So these are going to surround capillaries of the choroid plexus, and therefore it will limit the transfer of substances to and from the cerebrospinal fluid. It also allows the chemical composition of the blood and cerebrospinal fluid to differ. There are several breaks in the blood-brain barrier, and these are important for a variety of chemical exchanges to occur. So there are some in portions of the hypothalamus, and this allows for hypothalamic hormones that are created there to be introduced into the circulation. Also, we have some breaks in the blood-brain barrier in the posterior lobe of the pituitary gland. And this is going to allow for the hormones that are produced in the pituitary gland, like antidiuretic hormone and oxytocin, to be released rapidly into the circulation. The same thing goes for the pineal gland. Pineal secretions need to be allowed into circulation, and the choroid plexus needs to maintain the blood CSF barrier. The medulla oblongata is our most inferior part of the brainstem and is responsible for coordinating complex autonomic reflexes. So it's going to include 3 separate groups of nuclei, and these are going to control visceral functions. They're going to include sensory and motor nuclei of the central nervous system, as well as relay stations for communication in between the brain and the spinal cord. And as you can see based on its location, in the image on the right, it's that orangish sort of color that's just right above the spinal cord. So because it has that interface in between the spinal cord and the rest of our brain structures, you can see how it would have these broad, far-reaching responsibilities. Now we'll begin our discussion about the brainstem. We'll start with the medulla oblongata. The medulla oblongata also includes reflex centers that are responsible for autonomic and reflex activity. So the reticular formation is an area of gray and white matter that includes embedded nuclei within it. And this regulates autonomic functions, which include both largely cardiovascular and respiratory functions. So the center that is the cardiovascular center is going to include both cardiac and vasomotor centers. And this will include the control of blood flow throughout the peripheral tissues. So it can control things like vasodilation or vasoconstriction. The respiratory rhythmicity centers are just going to set the general pace for all of our respiratory movements. If you were looking at the medulla oblongata in a cadaveric specimen to identify the structures here in this image, you might have a difficult time because they're very subtle. In this image, you can see that everything is color-coded and labeled, and that makes it really easy. So let's take a quick look at this before we move on. So the medulla oblongata is going to have both sensory and motor nuclei for cranial nerves, specifically cranial nerves 8, 9, 10, 11, and 12. So what you can see here is there is a large green structure, and that's the inferior olivary complex, which we'll look at again in the coming slides. And then just inferior to that, you'll find that there's that red area, and also then there's that purplish rounded area. And these are the cardiovascular centers and the respiratory rhythmicity centers, respectively. Just in front of that in the image, you're going to see this grayish area, and that's just a general miscellaneous other nuclei. And so these centers here are responsible for both sensory and motor nuclei for those 5 cranial nerves we just discussed, which are 8 through 12. We also have just in front of that those 2 blue oblong areas, and that's the gracile nucleus and the cuneate nucleus. And these are going to take somatic information to the thalamus for processing. And lastly, we have the reticular formation, which is that long yellow structure. And the reticular formation is going to have nuclei and centers that are going to allow for regulation of our autonomic functions, and this extends into the pons and into the midbrain. Let's discuss some of those relay stations that have both sensory and motor pathways. So there's the gracile nucleus and the cuneate nucleus, and these are responsible for passing somatic sensory information to the thalamus. The tracts that leave the nuclei are going to cross over to the opposite side of the brain. It's something called the decussation of pyramids. And this is an important point to recall, that the tracts do cross over to the opposite side of the brain. There are solitary nuclei, and these are just going to receive visceral sensory information. And the inferior olivary complex is going to have the bulk of that complex creating the olives. And these are going to relay information about somatic motor commands. Continuing our discussion of the brainstem, we'll now discuss the pons. The pons is located immediately anteriorly to the cerebellum. So because of that, it's in a place where it will allow for the cerebellum to communicate with pretty much everything, either with the cerebrum superiorly, or the midbrain, or the spinal cord. So the pons has a really important role here. It contains sensory and motor nuclei of the cranial nerves, and nuclei that involve with respiration, such as the apneustic center and the pneumotaxic center. The nuclei and tracts that process and relay information here are sent either to or from the cerebellum, right? And the ascending, descending, or transverse pontine fibers here are also an important point. So take a look at this in the coming images. The transverse pontine fibers, pontine is just referring to the pons, right? These are axons that are going to link nuclei of the pons with the opposite cerebellar hemisphere. So these cross over in the same way that the decussation of the pyramids cross over as well. So here's figure 14.7 that I referred to in the previous slide. So you can see how the cerebellum is immediately posterior to the pons, and therefore the cerebellum needs to communicate with the pons before any of this information comes or goes either superior or inferior to it. So the cerebellum has descending tracts and ascending tracts in the pons that will carry motor commands and sensory information respectively to and from the thalamus and also the spinal nerves. We also have the transverse pontine fibers, which we just talked about, and these are going to connect the 2 cerebellar hemispheres, which are the right and the left. And then in the white matter, we have the apneustic and pneumotaxic centers, and these just adjust our respiratory rate. And then we have the reticular formation, which is that long orange structure that stretches out from the pons down through the medulla and even down further than that. And that's there to help us monitor our incoming sensations and also our outgoing motor commands. We also have nuclei of cranial nerves 5, 6, 7, and 8, but just only 8 in part. And so that will allow us to relay sensory information up to the brain and then inferiorly issuing somatic motor commands. And there are other nuclei and relay centers in there as well that both relay sensory and motor information to the cerebellum. So the pons is kind of a stopover station for the cerebellum. Now we'll wrap up the brainstem with a discussion about the midbrain. The midbrain has a number of different structures that are a little bit difficult to ascertain one from the next, but we're going to take a look at these in the coming slides, and I hope it should make sense. So some of the structures of the midbrain you should know are the cerebral peduncles, and these are nerve fiber bundles that are on the ventrolateral surface of the midbrain, and these include descending fibers to the cerebellum, which include descending motor command fibers. So let's take a look at figure 14.8 on the right, and the first thing you're going to see is that boxing, the corpora quadrigemina. We're going to look at that in the next slide, but this is just basically the 4 quadruplets. So anatomists were funny. I don't know how they named things, the things they did, but we're going to have to work with it. So we've got the corpora quadrigemina, and the 2 at the top are the superior colliculi, and the 2 at the bottom are the inferior colliculi. And then if you look at the base of the inferior colliculi, on each side we have sort of a base, and we call that the cerebral peduncle. And you can see there's a tiny little spot, by the way, just on that groove of the cerebral peduncle where the trochlear nerve exits. We also have the superior cerebellar peduncle, which is that broader base. And then you can see there's a space illustrated at the bottom, and that's the 4th ventricle. So that's the passageway of the cerebrospinal fluid. The tectum is just the roof of the midbrain, and it includes 2 pairs of sensory nuclei called the corpora quadrigemina, or the 4 quadruplets, again including the superior colliculi and the inferior colliculi. So the superior colliculi have visual centers, and the inferior colliculi have auditory centers. So I think of this in terms of superior has visual, inferior has auditory, because our eyes are above our ears. So that was easy enough for me to study and remember when I was in school. The tegmentum is just an area that's anterior to the cerebral aqueduct, and it has 2 different areas that are slightly different colored. So there's the red nucleus, which has a lot of different blood vessels, so that is what imparts that slightly reddish hue. And then the substantia nigra just literally translates to black substance, and it's composed of pigmented gray matter. And that black pigment that you see is actually melanin, which is a byproduct of dopamine production. Figure 14.8 shows you a cross-section illustrating where the tectum is, which is the roof of the midbrain, and that the tegmentum is just anterior to the cerebral aqueduct. So it's a little bit confusing, this image, so take a moment to take a look at this. But you'll see that the bulging that you see at the very cross surface of the image is the superior colliculus. Again, eyes over ears, right? So it's got visual centers there. And then let's look at the red nucleus and the substantia nigra here. So you can see the red nucleus is labeled on the left, and it's a very vague area. It's just slightly darker. It looks like it's part of a rounded circle. That's it. That's your red nucleus, composed of a lot of blood vessels. And then there's that gray-black area on both sides. You can see it's almost like somebody smeared ink or something across the surface. And that is our substantia nigra. Again, that black substance is there as a byproduct of dopamine production and is just melanin. Really, that's all it is. Now we'll move into section 14-6, the cerebellum. Let's look at some structures of the cerebellum. So the cerebellar cortex is very similar to the cerebral cortex. So we have gray matter, again composed of those neuron cell bodies, and that has a highly convoluted surface, right? So the image on the right is showing you the schematic, and then we actually have a real specimen on the right, giving you more of an idea of what it looks like. And so as you can see that there are lots of folds in the cerebellar cortex. In the cerebellum, we call these folia. Think of leaves like foliage. We use the tree analogy pretty extensively when we discuss the cerebellum here. So we're going to break the cerebellum into anterior and posterior lobes, and they're going to be separated by the primary fissure. So in the schematic on the left, you're going to see that that's delineated by gray showing the anterior lobe and more of a tan showing the posterior lobe. Whereas when you look at the image on the right of an actual cerebellum, it's much more subtle. You can see that there is that fissure that runs along horizontally there in the image. So we have 2 cerebellar hemispheres, the right and the left, and they are separated by the vermis, which is another name for worm, basically. And so that's just a narrow band of cortex that's going to separate the 2 cerebellar hemispheres right at the midline. We also have the flacco-nodular lobe, and that is just a location that's above the roof of the 4th ventricle. On cross-section, the cerebellum is beautiful. I mean, it truly is. It's got this amazing cut surface that looks like you're looking at a tree, which is why we call it the tree of life. So we have the Purkinje cell layer, and this is composed of large branched neuron cell bodies in the cerebellar cortex. And each of the cells here is going to receive input from up to 200,000 synapses. I mean, think of that. This is incredible. There's an amazing amount of processing that occurs here. The arbor vitae, again, the tree of life, is our highly branched internal white matter. Remember, the white matter is carrying all of our axons. And so the cerebellar nuclei are going to be those locations where we have the cell bodies, and they're embedded within the arbor vitae, and they will relate information to the Purkinje cells out at the cortex. There are tracts that are going to link the cerebellum with other structures nearby, like the brainstem, the cerebrum, and the spinal cord. And so these are called cerebellar peduncles. And I think of peduncle meaning stalk. Like if a polyp, for example, in the colon is pedunculated, then that means that it has a stalk, as opposed to if it's sessile, then it means it's broad-based. So the cerebellar peduncles, or stalks, are just those tracts. So we break them down into superior, middle, and inferior cerebellar peduncles. So as you can see in figure 14.9, we have the superior cerebellar peduncles that are going to link the cerebellum with the midbrain diencephalon and cerebrum, which makes sense, right? Because they're superior, so they're going to connect with things that are more superior. We have the middle cerebellar peduncles, which will carry communications in between the pons and the cerebellum, so that's more in the middle of the area. And then the cerebellar peduncles that will communicate with things going inferiorly, such as the medulla oblongata and spinal cord, are the inferior cerebellar peduncles. The cerebellum is important because it constantly adjusts our postural muscles at a very basic level. So it's going to fine-tune our movement of both our conscious and subconscious movements, taking in proprioceptive information to allow us to maintain an upright posture so we don't just collapse into a pile of goo. And so disorders of the cerebellum can create all sorts of problems such as ataxia, which will result in a disturbance of our muscular coordination. And this can involve us not being able to walk or sit upright so that we fall over. And that can arise from a number of different circumstances, ranging from damage to the cerebellum, from such as a trauma or a stroke or CVA, or it can also result from intoxication. But this is a temporary impairment, not a permanent one. I have a pop quiz question for you here, and this one is not something I would test you on because I would not expect you to be able to interpret an MRI, so I'll walk you through it. But the question here is, based solely off of the location of the lesion in this MRI, what clinical signs or symptoms might the patient have experienced? Now, I know you're not a radiologist, and neither am I, but let's take a quick look at this. What are we looking at? So we're looking at a cross-section of the brain. So in the image on the left, we have a section that's right through the center of the head, and you can see that towards the top of the image is the front of the face, and then towards the bottom of the image is the back of the head, okay? So we are looking here at the cerebellum, and that is occupying the majority of the inferior part of this image. So if you look at the anterior right part of the cerebellum, you're probably noticing this whitish area that is not on the other side. So hot tip, if you're ever pimped by a doctor who asks you, what are you looking at here? Always look for symmetry. If you're looking for things to be the same as on the right and on the left, that will usually give you the hot tip of what's different. So you'll see that that white spot on the right doesn't exist on the left, so that's your first tip that something's wrong. So now let's look over at the second image, which is giving you a different angle. So here we're looking at a cross-section through the brain, and so at the very top of the image is of course the top of the head, and at the bottom of the image is the bottom of the head with the person facing anteriorly. So in this case, what we're seeing here is again on that bottom right, we're going to see that whitish area that's consistent with that same whitish spot that we're seeing in that left image. This is a tumor, okay? So we know that the patient here has a tumor of some sort, which I'll talk to you about in a second. But based upon where it is, so we know that it's in the cerebellum, what signs or symptoms might occur from a lesion in the cerebellum? Would it be A, decreased urination from elevated ADH, B, irregular movements, or C, increased sex drive and inappropriate behavior? Okay, so this is interesting. So I'll give you a minute to think about it. Okay, so What could it possibly be? Could it be decreased urination from elevated ADH? Well, what would that have anything to do with? The pituitary gland is involved in ADH, but that's not what we're looking at here in the cerebellum. So what does the cerebellum do? Postural movement, right? So a regular movement would be your very best guess. So if somebody has ataxia, a regular movement, that would be consistent presentation with a lesion in the cerebellum. Now that's not to say that there can't be other complications and that presentations can't be very diverse when we look at brain diseases. But what we're looking at here is somebody who has a glioblastoma multiforme, which is a grade 4 astrocytoma, which is the most deadly of all the types of brain cancers, and people are typically dead within a number of months upon diagnosis of this. I actually know several people in my personal life who've had this, and also my favorite rock star, Gord Downie of the Tragically Hip, because I'm Canadian, he passed away from this several years ago as well. It's a very aggressive malignancy, and this is what it presents like in a rare case of a cerebellopontine presentation of a glioblastoma multiforme. Option C is increased sex drive and inappropriate behavior. Now, that we haven't discussed yet, but we will as we discuss other parts of the brain, specifically the frontal lobe. So let's move on. Section 14-7 in your text covers the diencephalon. Let's take a look. The diencephalon is responsible for integrating sensory information coming in with motor commands going out. So this includes the epithalamus, which is the area above the thalamus, the thalamus itself, and the hypothalamus. Hypothalamus just meaning the area below the thalamus. So the naming here wasn't terribly creative. We also have the pineal gland, which is the posterior aspect of the epithalamus, so it's more superior, and the pineal gland is responsible for secreting melatonin, which has an important role in maintaining our circadian rhythms and our sense of when it is day, when it is night, and when it is time to sleep. Interestingly, this is not quite adequate in people who are blind. If they are fully blind, they will still have problems sleeping sometimes, because even though there's melatonin produced and you would think that that would be indicating that it's time to sleep, our sense of vision and taking in daylight contributes to our understanding of when it is day and when it is night and how we secrete melatonin. So let's take a look at this image on the right. It's figure 1411 in your text, and you're going to see one major structure in the center that's broad and homogeneous and tan, and it's stretching out horizontally in the image, kind of curved. And that's the corpus callosum. And this area here is going to separate the two hemispheres of the cerebrum. We have the septum pellucidum, which will separate the right and left hemispheres. Inferior to that, we have the fornix. And I also want to walk you through just a couple other structures here. You can see the anterior commissure at the very front, that little area hanging down a bit. The parietal lobe is the primary part of the brain that we're looking at, the superior aspect. And then going down one step further on the list on the right, you'll see the choroid plexus is listed. And the choroid plexus is almost like a velvety surface in that area there. The thalamus is surrounding the third ventricle. And then we have the pineal gland, which is the tiny little rounded eminence just behind the thalamus. The hypothalamus is just beneath the thalamus. And then we have, as you can see going inferiorly, the cerebral aqueduct, which would carry CSF inferiorly. A little bit of the cerebellum is showing, and you can see on cross-section how beautiful that is. And then just inferior to the cerebral aqueduct, we also have the fourth ventricle. The thalamus is responsible for filtering and then relaying sensory information that's coming in from the spinal cord and from the cranial nerves, and then sending it up to the cerebral cortex for processing. So it's relaying information between those basal nuclei and the cerebral cortex. The third ventricle is going to separate our left and right sides, and we have something called the interthalamic adhesion, which is a projection of gray matter that's going to extend into the third ventricle on both sides. The diencephalon has thalamic nuclei, which are responsible for relaying sensory information to the basal nuclei and also to the cerebral cortex. There are 5 groups of thalamic nuclei, and these include the anterior, medial, and ventral nuclei of the thalamus. So the anterior nuclei are part of the limbic system, which manages our emotions. The medial nuclei of the thalamus are going to help provide an awareness of our emotional states, so we understand that we're actually unhappy, or that we are happy, or we're sad. And the ventral nuclei of the thalamus are going to take sensory information and relay it to the cerebral cortex for processing. So if you take a look at figure 1410, just over to the right, you're going to see an enlarged view of the thalamic nuclei. So on the left side here, you can see the pulvinar process sticking anteriorly to the right. And then we have some general sensory input being that purplish area that you see as part of the ventral nuclei. So the ventral nuclei will be sending information to the cerebellum. And then you can see that we have the anterior nuclei in orange kind of at the back there, and that's sending information to the limbic system. And then with the medial nuclei in red up at the top, which sends information to the frontal lobes, and that's a really important component as well. Of the 5 groups of thalamic nuclei, we also have the dorsal nuclei of the thalamus, including the pulvinar nuclei, which in the image on the right, you can see is the rightmost component of the thalamic nuclei, and the geniculate bodies, including the lateral and medial geniculate body. So the lateral geniculate bodies have visual centers, whereas the medial geniculate bodies have auditory centers, and these will affect our emotional states. The hypothalamus is the area just below the thalamus, and it extends from where the optic chiasm is, which is the area where the optic nerves cross over as they approach the brain, all the way to the posterior aspect of the mammillary bodies, which are shown in the image at the right as a rounded red area. The mammillary bodies have control reflexes that allow for us to eat and control those movements. And the mammillary bodies, I just have to point out, also have a funny name because mammary is referring to breast tissue, right? So basically this means that early anatomists looked at these rounded prominences on the brain and thought, oh, those look like breasts. I think that's kind of bizarre. But anyway, those are the mammillary bodies. So then as we look more inferior here, you can see that we have the pituitary gland hanging kind of inferiorly, and it's hanging by a stalk called the infundibulum. So the stalk will connect the hypothalamus to the pituitary gland. Also, we have the tuber cinereum, and this is just a swelling in between the infundibulum and the mammillary bodies. So it's just that little tiny space right there in between, and it's going to produce hormones that are going to affect how the pituitary gland operates. There are 8 primary functions of the hypothalamus. So one is to secrete antidiuretic hormone via the supraoptic nucleus and oxytocin via the paraventricular nucleus. So antidiuretic hormone obviously has importance in our urinary system, and oxytocin is important in both lactation and also in delivery of a child. The hypothalamus is also responsible for regulating body temperature via the preoptic area. It controls our autonomic function, coordinates our voluntary and autonomic functions, and coordinates our nervous and endocrine systems. So you can see this is a really important area. The hypothalamus also regulates our circadian rhythms, which is what is our day-night cycle, and it does that by the suprachiasmatic nucleus, and also has some conscious control of our skeletal muscle. So that's not when we go to reach for a cup of coffee, that's just the fact that we're sitting up and we kind of lean to one side, and you're not really thinking about how you're leaning to one side on the armrest. It's that sort of thing. It also helps us to produce emotions and behavioral drives through our feeding center, so that's our drive for hunger, or our thirst center, which is our drive for thirst, and our satiety center, which regulates our food intake. Now we'll look at section 14-8, the limbic system. The limbic system is not a physical structure, but it is a functional unit that works together to establish our emotional states. And it does this by linking our conscious functions of our cerebral cortex with the autonomic functions of our brainstem. In doing so, it facilitates our memory storage and retrieval as well. So if you look at figure 1412, you can see that there's a green area that's highlighted, and this is our cingulate gyrus, which is the superior aspect of the limbic lobe. And in it, there's a lot of different structures just below. We have the corpus callosum, which is separating the right and left hemispheres, the fornix, the pineal gland, the anterior thalamic nuclei, the hypothalamus, mammillary body, and also part of the temporal lobe of the cerebrum as well here. The parahippocampal gyrus is part of the inferior portion of the limbic lobe, and the hippocampus, which is part of the dentate gyrus. This is the posterior aspect of the limbic lobe, and this has a very important role in memory. The fornix, which you can see in the image above as a yellow broad stripe right in the center of the image, is a tract of white matter composed, of course, of axons of neurons, and it connects the hippocampus with the hypothalamus. The anterior nuclei of the thalamus are going to relay information between the mammillary body to the cingulate gyrus, which is that green area that you see in the image above. The reticular formation, remember that was that long structure that we saw in the brainstem, is responsible for regulating alertness, excitement, lethargy, and also sleep. So the limbic lobe of the cerebral hemisphere is going to include the cingulate gyrus, the dentate gyrus, the parahippocampal gyrus, the hippocampus, and the amygdaloid body. So the amygdaloid body, just by the way, it acts as an interface. So it works in between the limbic system and the cerebrum, communicating between them, and also various sensory systems. Now we'll discuss the cerebrum and its motor, sensory, and association areas. The cerebrum. This is the largest part of the brain, and again, what we think of as the brain. So it controls all of our conscious thoughts, our intellectual functions, and this is where we process somatic sensory information and motor information as well. So recall that the cerebral cortex is composed of gray matter because we have lots of cell bodies in that area, and there are also basal nuclei which have a lot of gray matter in them as well. The white matter is going to be deep to the cerebral cortex and is surrounding around those basal nuclei because that is where the axons of those neurons are located. So quick anatomy review. The gyri are those bulging convolutions of the cerebral cortex which increase the surface area for all of those cortical neurons. There is the longitudinal cerebral fissure, which is going to separate the right and left cerebral hemispheres. And then the four lobes of the brain are the frontal, parietal, occipital, and temporal. So the frontal, of course, is in front. The temporal is by your temples, so it's on the side. The parietal is up top and towards the posterior aspect. And the occipital is at the far back at the back of your head. Some additional structures of the cerebrum include the central sulcus, which is going to divide the anterior frontal lobe from the posterior parietal lobe. So you can see that located on the image highlighted just in between the red and the purple. The precentral gyrus of the frontal lobe is going to be that area that's highlighted in that pinkish red color, and it's going to form the anterior border of the central sulcus. The postcentral gyrus of the parietal lobe is going to be shown in the purplish area on your image at the right, and that forms the posterior border of the central sulcus. The lateral sulcus is going to separate the frontal lobe from the temporal lobe. So you can see that delineates in between the pinkish frontal lobe and that orangish temporal lobe just beneath it. The insula is also another name for island because it's insulated from the cortex. So it lies medial, so more towards the inner aspect, to the lateral sulcus. So you can't see it in this image here. And then the parieto-occipital sulcus is going to separate the parietal lobe from the occipital lobe. So you can see that in the image here, separating the parietal lobe from the occipital lobe. So how do the neurons in the cerebrum communicate with one another? They do so by association fibers, and these association fibers are going to form connections only within one hemisphere. So they're not crossing over to the other side. So association fibers will include both arcuate fibers and longitudinal fasciculi. The arcuate fibers are short, and these are just going to connect one gyrus with another. So one bulging area, one convoluted area to another. But the longitudinal fasciculi are longer, and they're going to connect the frontal lobe to other lobes, but again, these association fibers stay within the same hemisphere. They don't cross over to the other side. Sometimes, however, we have information that needs to cross over through different hemispheres. So in that case, we have projection fibers. So the projection fibers are going to link the cerebral cortex to the diencephalon, the brainstem, the cerebellum, the spinal cord, basically anything, but they're going to pass through the diencephalon if need be. The internal capsule is going to contain all ascending and descending projection fibers. Commissural fibers are bands of fibers that are going to connect the two hemispheres. So the commissural fibers you can see in the image on the right are shown in red. So they're crossing via the corpus callosum, and they're also crossing via the anterior commissure. Within the cerebrum, we have several basal nuclei, which are just masses of gray matter. Again, these are areas of neuron cell bodies that are embedded within the white matter, which is areas of the axons coming from the cerebral cortex and other areas, and these will direct subconscious activities. So some of these basal nuclei include the caudate nucleus, which you can see in the image below is shown in red. So these are described as having a large head, they're slender with a slightly curving tail. And then we also have the lentiform nucleus, which includes the putamen and the globus pallidus. The putamen is more lateral and the globus pallidus is more medial. So you can see those two other spots on both sides because these are bilateral structures. You can see the pale pink is the globus pallidus because it's more globe-like, it's more round. And then the one that's more regularly shaped is the putamen. And both of these lentiform nuclei are responsible for subconscious adjustment and modification of our voluntary motor movements. We also have the claustrum. The claustrum is just this thin gray stripe that you can see that's just lateral to the putamen, and it's just an area of gray matter right next to the putamen. This image here is showing you a superior view of transverse sections at different levels. So you can see there's one cut that's more superior, which is the top right part of the image, and one cut that is more inferior, that is at the bottom left aspect of the image. So starting off at the top, we have the corpus callosum, which is where we have those commissural fibers which allow communication between the two different cerebral hemispheres. There's the septum pellucidum, just inferior to that. So that is that thin-walled structure between the ventricles. So just lateral to the septum pellucidum on both sides are the lateral ventricles, the right and the left, and they are both the first and the second ventricles. Then there's the fornix. You can also see a cross-section of the third ventricle. And if you look down at the very bottom of the image, you can see that we have lateral ventricles shown as well. The basal nuclei are important for controlling our skeletal muscle tone on a subconscious level. So they help us with coordination of learned movement patterns like walking and lifting. So you're not thinking about walking when you're walking. You might be talking on your phone or pushing a baby stroller, and you're not really thinking about moving. You might think about starting to walk, but you don't think about actually continuing to walk. So our basal nuclei in our cerebrum help us with these structures and these movements. Parkinson's disease is one terribly serious disease which is caused by increased activity of the basal nuclei. So overall, what is the take-home? What does the cerebrum do? Well, the cortex of each cerebral hemisphere is going to receive somatosensory information from our body and then send the motor commands out to the opposite side of the body. And that's kind of a confusing thought, but the corpus callosum is there for a reason. It is there to help communicate information from one side of the cerebral hemisphere to the other side of the body. And it's definitely worth noting when you're thinking about the cerebrum and the brain that we like to think that specific areas of the brain have a very precise function, but this is actually not entirely true. It's close, but it's not perfect. So we like to say that this correlation is imprecise at best. Although I just said that So here's a caveat to what I just said. So although the correspondence between a specific function and a specific area of the cerebral cortex isn't quite totally precise, there are some generalizations that can be made. So the central sulcus is going to separate our motor and sensory areas. We'll look at images of this coming up in subsequent slides. So the motor areas are going to include the primary motor cortex as well as the pyramidal cells. So the primary motor cortex is at the surface of the precentral gyrus, and then the pyramidal cells are neurons of the primary motor cortex. The sensory areas are going to include the primary somatosensory cortex, and these will be identified at the surface of the postcentral gyrus. Let's take a look at figure 1416 for a second. So you'll notice first that we have the central sulcus separating the frontal lobe from the parietal lobe. And then you're going to see that there's bright red and bright blue right next to the central sulcus. And this is a very important point to take note of. So the central sulcus is separating the primary motor cortex, which is in bright red, from the primary somatosensory cortex in bright blue. Then fading off in faded red is the premotor cortex, and fading off in light blue is the somatosensory association cortex. I was trying to think of a way to make this relate to you in some way, and I don't know if you guys have seen the commercials with Motor, the guy that is half motorcycle and half man, but the motorcycle is in the front and then the man part is in the back. So motor is in the front, if that helps you at all. And then the movement, the somatosensory cortex, that's all in the parietal lobe, posterior to the central sulcus. So there are special sensory cortices here. The visual cortex is going to receive visual information, right? That makes sense. The auditory cortex receives information about hearing. The olfactory cortex receives olfactory information about smell. And the gustatory cortex is going to receive information from our taste receptors about what we're tasting. So you can see where these are located in figure 14-16 to the right. Association areas are just areas of the cerebral cortex that are going to take that incoming data and then coordinate some sort of motor response from it. So they're connected to both sensory and motor regions of the cortex because they need to both take in sensory information, make a plan, and then send out a motor response. So the premotor cortex is also known as the somatic motor association area, and it's going to be responsible for coordinating learned movements. Similarly, our sensory association areas will take information and process it as well. So now we're looking at the blue part of the image, mostly in the parietal lobe. So the somatosensory association cortex is going to monitor information and activity in the primary somatosensory cortex. And for example, one thing it will recognize is touch, how that feels, how you feel about it. The visual association areas will take information from the visual cortex and interpret that. And similarly, the auditory association area is going to take information from the auditory cortex and then manage that. Integrative centers are those that are involved with extremely complex processes. So these help us with things like doing math or speaking, writing, and understanding different spatial relationships. So these are restricted to either the left or the right hemisphere. And although we have, right and left anatomical symmetry, these different areas of the brain actually function very differently. And this is called hemispheric lateralization. So these integrative centers will receive information from these association areas. They direct the motor activities as a response, and they perform an analytical function of taking in that information and figuring out what the plan should be for motor activity to respond to something sensory. If you're taking an introduction to psychology course or an abnormal psychology course, you'll probably be discussing Wernicke's and Broca's aphasias. So Wernicke's area is where we comprehend language. So this is associated with our left cerebral hemisphere, and it's going to receive information coming in, sensory information from this space, and it's going to coordinate our access to visual and auditory memories. So if there's any damage to Wernicke's area, we won't understand what somebody is saying to us. Whereas with Broca's area, which is a motor speech area, the exact inverse is true. This is not an area of understanding, this is an area of speech production. So it's associated with the left cerebral hemisphere, and it regulates our patterns of breathing and vocalization. So somebody who may have had a cerebrovascular accident that affects Broca's area may have a very different outcome clinically than somebody who's had a cerebrovascular accident that's affected Wernicke's area. The prefrontal cortex of our frontal lobe is responsible for taking information from all of our cortical association areas, and then that allows us to perform abstract intellectual functions. What does that mean? It basically means that we can complete a game of three-dimensional chess in our mind. So we can see what the outcomes are or the consequences are of actions and think down the line longitudinally about multiple different outcomes for multiple different activities. This is not something that matures in us until we're actually in our mid-20s. So previously, in the mid-20th century, people who had mental illness were quote-unquote cured by a prefrontal lobotomy, which meant we would cut out a portion of the frontal lobe, specifically the prefrontal cortex, and that would eliminate the bad behavior that people thought was the problem with their mental illness. Of course, that did not cure anything. It just created a whole host of other issues. So the gentleman that you're seeing here on the right is Phineas Gage, who you will certainly learn about when you study psychology, physiology, or neurology. And this is a man who had accidentally had that iron spike that you see in his hand punctured right up through his face. So the image that you see at the bottom is showing you the way that that iron spike traversed through his head. After this happened, he had profound personality change and had a very different life. And he survived because it went through his prefrontal cortex and did not manage to hit any structures that would cause a massive bleed. So surprisingly, he survived, even though it was quite a long time ago, but it changed his personality and his social behaviors considerably. I really encourage you to look this up. Phineas Gage is a fantastically interesting story. Hemispheric lateralization, again, is just that there is a functional difference between the left and right hemisphere. Although we would think that the right and left cerebral hemispheres would function the same because they physically have symmetry, they actually have different functions on different sides. So the left cerebral hemisphere is going to be responsible for things like reading and writing and math. It's responsible for our speech, our language, and our ability to make decisions. Now, the right cerebral hemisphere is going to analyze sensory information, so things like touch and smell and taste and sight. All of those things are things we sense, and then the right cerebral hemisphere will interpret that information. The right cerebral hemisphere is also responsible for recognizing faces and voice inflections, so you can tell if somebody sounds angry or hostile or irritated or maybe sad when they're talking to you. To assess neural activity, one study that can be performed is an electroencephalogram, which is abbreviated as EEG. So an EEG will have electrodes that are placed either on the brain directly or on the skull, most typically, and then electrical patterns in the form of brain waves will be observed to see what is happening. Do we have normal activity or do we have abnormal activity, or potentially even absence of activity in dead brain tissue? So the types of typical brain waves we would see are alpha waves, beta waves, theta waves, and delta waves. Let's look at those in the next slide. As you can see, each of these 4 different types of waves have a very different appearance. So starting from the top, you can see alpha waves. And alpha waves are what you would expect in adults who are just sitting still, relaxing, with their eyes closed. So these waves will disappear when people are sleeping, but they'll also disappear when people start to focus on a very specific task, at which point that's when you'll start to see beta waves. So the beta waves are much more jerky because we've got somebody who's paying attention, focusing very seriously on something, or potentially under stress. Theta waves are brainwaves that are seen typically in children and also in adults who might be extremely frustrated. I think we all know that when we're feeling very frustrated with something, it's really hard to focus and think clearly. So for example, if you're really mad and really upset about something and you try to write an email, you know that's not going to be your very best email that you could possibly write because you're worked up. And that's just the way pretty much everybody is because that's a biological response. My suggestion, by the way, is if you're ever really emotional about something, take a minute and breathe if it's not an absolute emergency, and you'll find that you can respond to almost any threat or any problem a little bit better because biologically you will actually have these theta waves relax down into maybe a beta wave so you can think seriously about how you want to move forward on something. Delta waves are going to be very different. You can see that these are these big, wavy, irregular structures, and these occur when we're deep asleep, and also they're present in certain pathological conditions. To allow for synchronization of electrical activity between the right and left hemispheres of the cerebrum, we have a pacemaker mechanism. And if this becomes broken in any way, then we have a desynchronization. And so that can arise from a situation where there's an injury or a tumor. So for example, seizures are basically a temporary cerebral disorder because we don't seize indefinitely, right? And so this will be accompanied by a change in electrical activity, which is why when somebody is seizing, we may hook them up to an EEG so we can see what part of the brain is having the seizure disorder. So the symptoms that the person is experiencing will depend on what area of the cerebral cortex is affected by the seizure activity. As a section of 14-9, we're now going to review all of the cranial nerves. There are 12 pairs of cranial nerves, and these nerves are connected to the brain, as opposed to spinal nerves, which attach to the spinal cord. So we're going to classify all of these 12 sets of cranial nerves as being either primarily sensory, special sensory, motor, or mixed. So the primarily sensory are going to be those that are carrying somatic sensory information. So that's things like touch, pressure, vibration, temperature, and pain. In essence, primarily sensory means anything that you might detect with your skin. Whereas special sensory is going to be those things that are sensed through our special senses. So our special senses are things like smell, sight, hearing, balance. So in other words, things that we sense through our head, through our face. The motor aspect are going to be axons of somatic motor neurons, and cranial nerves that are mixed will have both sensory and motor fibers. Cranial nerves are going to be classified firstly by what their primary function is, but they can also have very important secondary functions as well. In fact, several cranial nerves will distribute autonomic fibers to peripheral ganglia as well. As we discuss the cranial nerves, we're going to keep looking at this image so you can keep track of how these cranial nerves relate to the brain itself. So this is the view from the inferior aspect of the brain. So the top of the image is the anterior surface of the brain at the frontal lobe, and the bottom of the image is the posterior aspect of the brain at the inferior aspect. So you can see the spinal cord at the very bottom and then the cerebellum just next to that. So let's look at all of these different cranial nerves, keeping in mind the structures are bilateral. So at the very top, the very first thing you see labeled on the right is the olfactory bulb, which is the end or the termination of the olfactory nerve, which is cranial nerve 1. You'll note that we use Roman numerals to identify all of these different structures. The optic nerve is number 2, and so cranial nerve 2 is responsible for our eyes and our vision. Oculomotor nerve is 3, so the oculomotor is broken down into oculo, eye, motor, that's movement, so it's responsible for moving our eye. The trochlear nerve is number 4. Trigeminal nerve is the 5th cranial nerve, and the trigeminal nerve you'll see has tri in the word, which means it has 3 of something, 3 branches. Abducens nerve is number 6. The facial nerve is number 7, so it innervates most of our face. The vestibulocochlear nerve is 8, so it innervates the vestibule and the cochlea. Those are both structures in our ear. The glossopharyngeal nerve innervates the tongue. Glosso is tongue, and pharyngeal is throat. The vagus nerve is number 10, and that is a wanderer, which we'll discuss later on. And then the last 2, the accessory nerve is 11, and the hypoglossal nerve is 12. If you were just thinking in the previous slide, how am I ever going to remember all of these cranial nerves? There's 12 of them. How am I going to keep them in order and what they're for? Here's a trick. A mnemonic. A mnemonic is going to take the first letter of these words that you see here below, and it correlates with the first letter of the cranial nerve. So that way you can match them up and keep them in order in your mind, if only you can remember this mnemonic. So there are all of these different options for you to think about. I learned a different one, which was rather inappropriate when I was in graduate school, which I still recall and I can't share with you here. So if these do not seem to suit your fancy, you are more than welcome to research other options for mnemonics for cranial nerves. But I think probably the very best one is the third one listed here, which is O-O-O, to touch and feel, very good velvet, ah. And so keep in mind the ah at the end is A-H. So the A is going to be the accessory nerve and the H is the hypoglossal. So the ah has the last 2 cranial nerves together. Another mnemonic that we learned in grad school was how to remember cranial nerve function, because we're going to go through all of these different nerves and what they're for, and some things will be easily apparent and you'll be able to remember, and some things are going to be a little bit more difficult. So when we're discussing the function, whether we have a nerve that's got a stronger sensory role, a stronger motor role, or both, / mix, same thing, we can use this mnemonic. It says, some say marry money, but my brother says big brains matter more. So you might imagine this is also something that can be made to be very inappropriate. That is also what I learned in graduate school, and you will probably hear when you're in nursing school. But for now, take this, use this, and it will help you. So the S, of course, is sensory, the M, of course, is motor, and then the B is both or mixed. So there are a couple caveats to these mnemonics here, though. So any nerve that has motor function also has proprioception, which could be sensory, but that's assumed and not part of this overall mnemonic schematic. So cranial nerves 3, 7, 9, and 10 have parasympathetic fibers, and cranial nerve 5 has all of the sympathetic fibers for the head. First off, let's look at the olfactory nerve. So this is cranial nerve 1. The primary function of the olfactory nerve is for smell, right? This is a special sense, and it originates in the receptors of the olfactory epithelium. So it'll pass through the olfactory foramina in the cribriform plate of the ethmoid, which we'll take a look at in the subsequent slide, and the destination will be the olfactory bulbs. So that's located on either side of the crista galli, and the olfactory tracts are going to carry that information with axons of postsynaptic neurons, which will lead to the cerebrum. Figure 14-19 here will show you what we're looking at when we discuss the olfactory nerves. So you can see the yellow structures in the very center of the image. So you can see that there are those thin thread-like structures extending down, and those are olfactory nerve fibers that are responsible for sensing smell. So they're going to pass that information up through the bony tissue, up through that cribriform plate of the ethmoid sinus, into the left olfactory bulb, which is the end or termination of the olfactory nerve, which then will send information through the olfactory tract to the olfactory cortex in the cerebrum. Cranial nerve 2 is the optic nerve. So the optic nerve, of course, is a special sensory nerve that's responsible for vision. So it originates in the retina of the eye and then passes through the optic canals of the sphenoid sinus. And it arrives as destination, which is the diencephalon, through the optic chiasm, which is the point at which it crosses over. So if you look at figure 1420 on the right, you're going to first locate the optic nerve, and you can see it's transected. And then you'll see that it kind of has a U-shaped area where the other side's optic nerve is also transected. That area where they meet, that is the optic chiasm. So the optic chiasm is where the optic nerves converge, both the right and the left, and that's the point at which some fibers are going to cross over onto opposite sides of the brain. The optic tracts are going to reorganize those axons, and that continues on to the lateral geniculate body of the thalamus. So you can see in this image here that the eye on the right has blue fibers and the eye on the left has red fibers. So they travel along through the optic nerves on both sides, and then you can see as those meet at the optic chiasm, you can see that now at that point on in the optic tract, you can see that you have both blue and red fibers. So that's where we have the crossing over point at the optic chiasm. So the optic tract will carry things to the lateral geniculate body, which is in the thalamus, and then that will head to the visual cortex, which is in the far back of your head in the occipital lobe. The oculomotor nerve is cranial nerve 3, and its primary function is motor because oculo, eye, motor is movement, right? So it's responsible for eye movement. This originates in the midbrain, and it passes through the superior orbital fissures of the sphenoid, and then each of these oculomotor nerves is only responsible for controlling 4 of the 6 eye movement muscles. So there are still the 2 others that require other movement. So the oculomotor nerves will deliver autonomic fibers to the neurons of the ciliary ganglion, and these will control intrinsic eye muscles. The trochlear nerves are cranial nerves 4, and their primary function is motor, again for eye movement. So this is picking up for those 2 other eye muscles that weren't covered by the oculomotor nerve. So they originate in the midbrain and then pass through the superior orbital fissures of the sphenoid, with the destination being the superior oblique. Figure 14-21 shows you an image of the eye and all the different muscles that are responsible for moving the eye. But if you look at the very center of the image, you're going to see a very wide, broad, bright yellow structure, and that's the optic nerve, which is our cranial nerve number 2. Cranial nerve number 3 is the oculomotor nerve, which is extending from the back of the image at the far right at the top down towards the eye itself. The trochlear nerve, again, remember it has responsibility for 2 of the 6 eye muscles. So it is coming along the top of the image, extending up towards the superior oblique muscle of the eye. Before we discuss the trigeminal nerve in more detail, I wanted you to have a moment to take a look at figure 1422 from your text to see how the trigeminal nerve branches out. So you can see that we have that semilunar ganglion, which is the large rounded area in the trigeminal nerve. And then that branches off to three different areas: the ophthalmic, maxillary, and mandibular. And it's exactly what you think. So the ophthalmic branch is going towards the eye. The maxillary branch is going towards the cheekbone, and the mandibular is going down along the jaw. So the trigeminal nerves are cranial nerve 5, and they have the primary function that is mixed. So it's both sensory and motor of the face. So on each side, they're going to pass through, for the ophthalmic nerve, the superior orbital fissure. On the maxillary nerve, it will pass through the foramen rotundum, and on the mandibular nerve, it passes through the foramen ovale. Of course, the origin varies contingent on which branch we're discussing as well. So the origin of the ophthalmic nerve, which is sensory, is going to be through the orbital structures in the nasal cavity. So it will innervate the skin of the forehead, the upper eyelid, and the eyebrow, and also a part of the nose. As you can see how it crosses over and just goes down across the very top bridge of the nose. The maxillary nerve is sensory, so it innervates the lower eyelid, the upper gums, lip, and teeth, also the cheek and nose, the palate, and part of the pharynx. So this does basically the middle of your face. The mandibular nerve is broken down into two components, the sensory and the motor components. So the sensory components innervates the lower gums, teeth, and lips, the palate, and part of the tongue. And the mandibular nerve will send information from the motor nuclei of the pons. The destination of the trigeminal nerves is in sensory fibers, specifically the sensory nuclei in the pons. The motor fibers of the mandibular nerve will have their destination be the muscles of mastication, which is just the muscles that allow you to chew, because mastication is chewing. These are the largest cranial nerves, and they have those three major divisions. So there's the semilunar ganglion, which contains the cell bodies of the sensory neurons. Tic douloureux, which is pronounced douloureux en français, which I actually speak a little bit of because I was raised in Canada, is also known as trigeminal neuralgia. And this is a condition where people have a really extremely painful maxilla and mandible. Fun story for you. When I was on my first trip with my husband when we were dating, we went to Costa Rica, and I had a fractured molar, which was exquisitely painful, and I couldn't get adequate pain medication where we were because we were in a very rural part of the country, and I would end up crying and drooling profusely occasionally. And I mean, that's embarrassing when you're first dating somebody. My husband was fairly convinced that I had tic douloureux, and there wasn't too much to do about it until I got back home and then figured out that the tooth had to be pulled. So that's my fun story about tic douloureux for you, which affects the trigeminal nerve. The abducens nerves are cranial nerve 6, and the primary function of the abducens is motor for eye movement. It originates in the pons because it's sending information from the pons to the eye. So the information passes from the pons through the superior orbital fissures of the sphenoid and arrives at its destination in the lateral rectus muscles of the eye. Before we discuss the facial nerve, let's take a look at how it branches, because like the trigeminal, the facial nerve branches pretty considerably. So the branches of the facial nerve are the temporal, zygomatic, buccal, marginal mandibular, and cervical. So the temporal goes up to your temples, zygomatic goes towards your cheeks, buccal goes towards your mouth and lips, the marginal mandibular goes towards your lower jaw, and the cervical goes down towards your neck. The primary function of the facial nerves, which is cranial nerve 7, is mixed, so it's both sensory and motor. So it originates, for the sensory purposes, on taste receptors on the anterior 2/3 of your tongue, and the motor aspect is going to originate on the motor nuclei of the pons. So this passes through the internal acoustic meatus to the stylomastoid foramina. The destination of the facial nerves, if sensory, will be the sensory nuclei of the pons, If somatic motor will be the muscles of facial expression, and if it's visceral motor will be the lacrimal gland, which allows for us to tear and cry, and the nasal mucus glands, as well as the submandibular and sublingual glands of the mouth. 3 facial nerve structures include the geniculate ganglia, the pteropalatine ganglia, and the submandibular ganglia. The geniculate ganglia will contain cell bodies of sensory neurons. The pteropalatine ganglia will have postganglionic fibers, and these are there to innervate the lacrimal glands, which allow for you to tear, and glands of your nasal cavity as well as your pharynx or throat. And the submandibular ganglia will innervate the submandibular as well as the sublingual glands of your mouth. So a disorder that is characterized by inflammation of the facial nerve is called Bell's palsy, which you can see illustrated in the image at the right. And this is typically due to a viral infection, and the signs and symptoms that accompany this include paralysis of the facial muscles on the affected side, so it's typically one side only. And you'll also have a loss of taste sensation on the anterior 2/3 of the tongue as well. Now typically these symptoms will fade after weeks or months, but sometimes even sooner. Another fun story for you, not that this is all about me, but just to help you remember this, is how my facial nerve was clearly impaired back when I was 8 years old, and I had my first migraine headache right on the day before my mother's wedding to my stepfather. And so I was a very charming looking little child with a very bad perm and a very awkward pale pink, like bubblegum pink dress, and half my face was drooping. So the day before my mom's wedding, she took me to the hospital, to the emergency room, because she wasn't sure if I was having a stroke or what was going on. But I did not have a cerebrovascular accident. What I did have was a viral infection and a Bell's palsy, and it did pass, but the photos are awful, and I considered showing you a picture here today, but I passed on that because I thought you don't need to see that. All right, let's move on. Figure 14-23 shows you the origin and the branches of the facial nerve, but what I'd like you to focus on in this image is the smaller image at the top right, which shows you the distribution of those nerves and how it innervates the majority of the face. So you can see in blue how the temporal branches are covering part of the eye, and then the zygomatic branches are going to cover the majority of the cheek and nose. And then there's that small strip of the buccal branches because that heads towards the lips and mouth. And then the marginal mandibular branch hits your jaw, whereas the cervical branch, of course, is going to be innervating more of your neck. Before we look at the vestibulocochlear nerve, I want to show you how this actually works in your ear. So the large yellow structure that you see in figure 14-24 is the vestibulocochlear nerve. And you can see that it branches to 2 distinct places. One is the vestibular nerve, and the other is the cochlear nerve. And so the cochlea is actually our organ of hearing. So I'm sure you've heard of people having cochlear implants, and that is to allow people who cannot hear to actually hear. You do not hear of people having vestibular implants because that's a totally different function altogether, which we're going to learn in the lab, and you are going to love, by the way. But the vestibular nerve is there for our sense of balance. So you can see that the vestibular nerve approaches those 3 semicircular canals, which are important in determining our sense of, even if you close your eyes, you know if you are right side up or upside down. That is thanks to your semicircular canals and your vestibular nerve. So the vestibulocochlear nerve has branches of both the vestibular nerve for balance and equilibrium and the cochlear nerve for hearing. The vestibular cochlear nerves, which are cranial nerves 8, has the primary function, of course, of both balance and equilibrium through the vestibular nerve and hearing through the cochlear nerve. So the vestibular cochlear nerves are going to originate in the receptors of the inner ear, as we just saw in the previous image, and they'll pass through the internal acoustic meatus of the temporal bones and arrive at their destination, which is the vestibular and cochlear nuclei of the pons and the medulla oblongata. So again, the vestibulocochlear nerve structures are the vestibular nerve and the cochlear nerve. The vestibular nerve is going to originate at the receptors of the vestibule, which allow for our balance and our equilibrium, and it will connect to the vestibular nuclei of the pons and medulla oblongata. The cochlear nerve is going to monitor receptors in our cochlea to allow us to hear, and that's going to connect with cochlear nuclei in both the pons and the medulla oblongata. The glossopharyngeal nerves are cranial nerves 9. And the primary function is mixed, so that includes both sensory and motor functions for both the head and the neck. The origin for the sensory component is the posterior one-third of the tongue, and part of the pharynx, palate, and also the carotid arteries. The motor origin is the motor nuclei of the medulla oblongata. So keep in mind, because the glossopharyngeal nerve innervates the sensation of the posterior one-third of the tongue, if you were to have a Bell's palsy, this part would not be affected. Why? Because Bell's palsy affects only that of the facial nerve, not of the glossopharyngeal nerves, so the posterior one-third of the tongue would be unaffected. The glossopharyngeal nerves are going to pass through the jugular foramina between the occipital and temporal bones, with the destination for the sensory element being the sensory nuclei of the medulla oblongata. For somatic motor, the pharyngeal muscles which are involved in swallowing, and for the visceral motor, the parotid gland, because remember, somatic motor is something that we theoretically can control, whereas the visceral is not. So you cannot make your parotid glands salivate and produce any secretion upon will. But you can, however, force yourself to swallow if you like. So that would be the difference between the two here. Some glossopharyngeal nerve structures to know include the superior ganglion and inferior ganglion. So you can see in the image at the right, in the broadest part of the glossopharyngeal nerve, there is one slight dilatation, that is the superior or jugular ganglion, and then the one that is more inferior is the inferior petrosal ganglion. If you look just over to the left of that, you're going to see the optic ganglion, and this is where the visceral motor fibers are going to synapse. The vagus nerve is cranial nerve number 10, and its primary function is mixed, is both sensory and motor. And when I say that it is mixed, it's because it is distributed throughout the thorax and abdomen, and it wanders everywhere. The origin for the sensory component includes part of the pharynx, the auricle and external acoustic meatus, the diaphragm, and the visceral organs in the thoracic and abdominopelvic cavities. The motor origin is the motor nuclei in the medulla oblongata. This passes through the jugular foramina between the occipital bone and the temporal bones. But you're going to notice here that I just said the diaphragm, and that seems like that's pretty far south of where we were talking about before. We've previously discussed all these cranial nerves as innervating mostly our face, head, and neck. The diaphragm, by the way, is what's affected when we get the hiccups. So when you get the hiccups, it's because your vagus nerve has actually been irritated. The destination for the sensory component of the vagus nerve is the sensory nuclei and also the autonomic centers of the medulla oblongata. For the visceral motor component of the vagus nerve, the destination includes the muscles of the palate and pharynx and the muscles of your digestive, respiratory, and cardiovascular systems in the thoracic and abdominal cavities. So basically, like I said, that vagus nerve, it wanders everywhere. As you can see in figure 14-26, you can see the vagus nerve illustrated in yellow, and look where it goes. It goes everywhere. It goes to your heart, to your lungs, your diaphragm, to the stomach. It goes down to the lower intestinal tract. It goes everywhere. So our vagus nerve, truly a wanderer. When I discuss the vagus nerve as a wanderer, it is because it branches and radiates extensively. So the superior ganglion and the inferior ganglion are those which both contain sensory neurons. So figure 14-26 shows you what that looks like. So you can see the superior ganglion of the vagus nerve as well as the inferior ganglion of the vagus nerve zoomed in on at the right, and then at the left image you can see what it looks like more in relationship to the face and neck. The accessory nerves are cranial nerves number 11, and the primary function of the accessory nerves is motor to the muscles of the neck and the upper back. The origin is going to be the motor nuclei of the spinal cord and the medulla oblongata. So these nerves are going to pass through the jugular foramina in between the occipital bone and the temporal bones, arriving at their destination, which is either the internal branch or external branch. The internal branch will affect the voluntary muscles of the palate, pharynx, and larynx, and the external branch will affect the sternocleidomastoid muscle, which is this incredibly long muscle in your neck, and the trapezius. Accessory nerve structures include the spinal root, and the spinal root is going to contain motor fibers that start off in the anterior gray horns of the first 5 cervical segments of the spinal cord. The cranial root also has motor fibers, and these start in the medulla oblongata. The internal branch is going to join up with the vagus nerve, and that together will innervate voluntary swallowing muscles as well as muscles of our vocal cords. And the external branch is going to control our sternocleidomastoid and trapezius. The last cranial nerve is hypoglossal, and that is cranial nerve 12. The primary function of the hypoglossal nerve is motor for tongue movement. Again, hypo meaning under, and glossal, tongue. So it originates in the motor nuclei of the medulla oblongata, passing through the hypoglossal canals of the occipital bone, finally arriving at its destination, which is the muscles of the tongue. Lastly, let's discuss section 14-10, cranial reflexes. We're going to discuss cranial reflexes later on, but for now it's important to note that there are monosynaptic and polysynaptic reflex arcs, remembering of course that monosynaptic ones are much quicker because there's only one synapse, whereas if there's multiple synapses it's going to take more time. These will also include both sensory and motor fibers of the cranial nerves, and these are important because we can use these cranial reflexes to check to see if these nerves are functioning properly as part of a neurological workup. So as you can see in table 14.5, we have a number of different cranial reflexes that will show you what exactly should happen if that specific reflex is working and what specific nerve that relates to. So let's start on the far right. You see blinking of eyelids. If you can blink your eyes, if something touches your cornea, that's your corneal reflex. And so we know then the trigeminal and facial nerves are working properly. Similarly, we have a tympanic reflex. So if you hear a very loud noise, then you would respond in a very specific way. Auditory reflexes are the same sort of situation in which you would have an eye movement and or head movement triggered by a sudden sound. So if somebody were to clap their hands right before your right ear, you're naturally going to turn to look to your right to see what was that sound. So that would indicate your vestibulocochlear nerve as well as a whole variety of other cervical nerves. 3, 4, 6, 7, 10, and cervical nerves would be working properly. To be able to determine whether the vestibulo-ocular reflexes are working properly, we'd want to be able to see if somebody could rotate their head properly, because the opposite movement of the eyes to stabilize field of vision would be an important thing to note. There are other reflexes that are visceral, the pupillary reflex and the consensual light reflex, and we're going to work on these in the lab, and they're a lot of fun. These are just a very quick review of some cranial reflexes, and this by no means is meant to be an exhaustive idea of what is involved in a neurological exam, but just to give you an idea of how some of these reflexes can indicate how the performance is of these specific cranial nerves. Thank you everybody for your attention. I hope you enjoyed today's lecture. I think it's super fun. The course just continues to get more fun from here. So get ready. The next time we're going to get together will be for chapter 15 when we study the sensory pathways and the somatic nervous system. I hope you have a nice week. These are the references that I've used for today's lecture. You can see, of course, that the first thing I've listed is our textbook. And then below that, you can see I've listed a number of different things that I've used to augment the lecture to show you some clinically significant applications of what we're studying in class today. I hope you like them. I really think you should check out Phineas Gage, by the way. It's a really fascinating story.